Welcome, everyone. Thank you for joining us today for this webinar on our new software, Multi-Channel Suite. My name is Carla Bellack, and I'm the marketing manager here at Multi-Channel Systems. Before we get started, I wanted to point out two things. First of all, we are recording this webinar, and we will send you the link to the recording during the next days. On the right-hand side, you see a Q&A window. Please make use of the window, type your question in there. Only myself can see the question, and we will have a Q&A session after the presentation, and I will read out your question to today's speaker. Today's speaker is Dr. Frank Hofmann. He is an application scientist here at Multi-Channel Systems. He earned his PhD in biology at the University of Tübingen and joined our company in 2007. Today, he will give a presentation of about 30 minutes, and afterwards, he's available for the Q&A session. So I will hand over to Frank to get started on the presentation.
Hello, everybody. Thanks for joining us today. Today's topic is the new software, the Multi-Channel Suite. So first about the motivation. Why do we do this? I mean, we have the MCREC software, which is running fine, and we have more focused software packages. So why do we get through all this trouble and design a brand new software? The reason is very simple. The MCREC software is, in terms of software ages, ancient. It's more than 15 years old, and during the years, there have always been issues coming up that we simply could not solve in MCREC because it is simply impossible. And also the file structure is very outdated. So we decided at some point, okay, it doesn't make sense to add up more and more stuff to MCREC, but we will start from the scratch and design something completely new, which is also more user-friendly, more modern, and easier to use all in all. So also, I have to point out that this software is still under development. So, of course, it is not the final stage that you will see when you download the software today, but it's an intermediate stage. And the idea was to get the software out in the field and get some feedback from customers to see how we can improve and what functions should be added. It is meant to be a general purpose software, not something which is application focused like the Cardio 2D software or the LTP Director. And in such, it is an alternative to MCREC. So far, it has not as many features as MCREC, of course, but we will add up features until we are at the point that they are more or less equivalent in functionality, and then we will add up. To begin with, we focused on the recording and the data export of the software, and the analysis part is still lagging a bit behind. In contrast to MCREC, the Multi-Channel Suite consists of 3 different software modules, one for recording, one for data analysis, and one for data export. And as I said, recording and data export are relatively advanced. Analysis is lagging a bit behind. An immediate benefit that you would have from using the Multi-Channel Suite over MCREC when you have an MEA 2100 system is that the hardware of the 2100 is capable of doing a 24-bit AD resolution. However, the MCREC software limits us to 16-bit, but now with the Multi-Channel Suite, you can benefit from the full 24-bit AD resolution with the 2100. The Multi-Channel Suite has an easy drag and drop interface, and it's very clear where the data goes during in that software. That is something that always confused new customers when they came in contact with MCREC. And just like MCREC, the software is free of charge. You can download it from our software right now, and it runs with MCS devices only. That's why we can afford to do that. And a question which came up into this morning's seminar was what the operating system should be, and it runs only on Windows 8. That's one compromise we had to make because it's much more complicated, of course, to keep a software compatible with many different operating systems than just one. So a quick overview of the 3 different parts of the software. The Multi-Channel Experimenter, which is the online recording software, currently supports the 2100 MEA system, the wireless system, the old one and the new one. and just recently added the older USB ME data acquisition system. So if you have an older MEA 1060 data acquisition, MEA 1060 MEA system with an ME data acquisition system, a USB-based ME data acquisition, you can also use the multi-channel suite. Just like in MC-REC, one instance of the software can be opened for each individual data input that you're using. So if you have an amplifier with many different MEAs at the same time, you will open the software as many times as you have MEAs running, or as many times as you have inputs to your ME data acquisition system, and control each individual data input with one independent instance of the software. From all the virtual instruments that are inside the software, the data acquisition, the stimulator, and the recorder can open only once. Any other instrument you can open as many times as you like. They are simple drag and drop. You will see that in a minute. And one very important feature, I think, is that custom plugins are possible. That means that we will provide a developer toolkit. So you can make your own instrument, program it with either an analysis algorithm of your liking, or also you can make a hardware interface, like for example, a pump control or something like this, and incorporate it here in our core software. You just need to stick to certain limitations concerning the data coming in and the data going out, and then you can simply use it here. Our hope is that we will have in the long run sort of a community where people share their custom applications. The multi-channel analyzer is the offline analysis software. So far, as I said, this is a bit of a weak spot. There are not as many functions available right now, but one very interesting feature, I think, especially for people doing in vivo work in behaving animals with a wireless system, is that you can synchronize the electrophysiological data with a video stream. And so you can have a look at the video and at the same time record the electrophysiological signals, and then look at both of them synchronized, and you can jump back and forth. So you can look for a certain spot inside your video and jump immediately to the corresponding electrophysiological data and vice versa. And here are also custom plugins possible. Last but not least, not the last important part of the package, is the multi-channel data manager. The data manager is for data export to third-party applications. It supports all file generated with MCS software. So whatever you use, whether it's an MCD file, Cardio 2D file, or from the CMOS system, from the multi-well system, or from the multi-channel suite, doesn't matter. The data manager will read it. You will have an overview of all available data streams within that file. You select the data streams that you need, and then with one single mouse click, you can export them directly to HDF5, which is compatible with MATLAB and Python. And you can also export them to NeuroNexus format NEX and to Spike2 format CED. And if you use the HDF5 format, there's also a data compression included. So it's very straightforward, very fast, and much easier to use than the NeuroShare interface that we used to have to export stuff to MATLAB. And the general idea was to get the manager, to prioritize the manager for people to be able to record with the new software and do the analysis in the software of their liking anyway. And so we prioritized our efforts to these two parts of the software package. So let's begin with a short movie of the experimenter. Here you go. The multi-channel experimenter, the online tool, comes up like this. You have here your virtual instruments. You get a representation of all attached data sources here. In that case, it was an USB ME256 with 4 different inputs, A, B, C, D, and you get one icon for each of them. So you drag that icon over. Again, we have here another data acquisition system attached, the MEA2100. You see, you could get not only the name of the device, but also a little icon. In case you have a 2 times 60 Hz stage, you can even see from the icon which of the 2 MEAs you're addressing with that specific icon. So it makes things much easier. And here are the other virtual instruments which are available at the moment. And the movie will show you how that simple drag and drop interface works. So you simply take the device, drag it over. The recorder, maybe a spike detector that detects spikes. You want to filter your data before you detect spikes, you put in the filter, and now you connect the devices. You see that the inputs and outputs of the device are color-coded, and only matching colors can be connected. And when you click on one icon, you will get a tab on the other hand with the control functions for that specific instrument. So data acquisition, filter, spike detector, and recorder. So it's pretty straightforward. And already from this tree here, you can see how the data flows. It comes out here. These on the downside are always the outputs of a device. On the upper side are the inputs. The electrode raw data labeled in blue comes out from the data acquisition, goes into the recorder directly, unfiltered. At the same time, it goes through the filter. The filter data goes through a spike detector. The spike detector detects the spikes. Its output are not raw data anymore, but spike cutouts and timestamps, and these also go into the recorder and then show up here as 3 individual data streams. And just by looking at this, you will see immediately what is being recorded. There's not so much room for confusion as there used to be in MC-REC. So, okay. Let's go to the next. So I will go now through the different instruments, but first let me show you what happens when you have more than one instance open. These are 2 instances of the software. You see here, I have dragged in the USB ME data acquisition, and over here I've dragged in the 2100 system. So in this instance, you see in light blue labeled the data acquisition which is actually in use in that rack. The other 3 which theoretically could be used are darker blue, and the one which is already occupied by a different application, this one, is gray. So this one can't be used, this one could be used if you remove that one first. The same goes over here, so it's easy visible what is possible and what not. So it cannot be that you connect the same device to 2 different instances. So if you wanted to use them all at once, you could open 5 times the software. That would probably be a bit much for one computer to handle, but in theory it's possible. Okay, so let's first go through the different data acquisition systems. You drag in the data acquisition system that you want to use, click on it, and then you get the control functions over here. There are the basic controls, like you set the sampling rate, you set the gain, which is necessary for the ME recording, for the ME data acquisition systems, not for the 2100, where the gain is detected automatically. You can define whether you want to record the additional analog channels, and also you can have a look at the hardware information. And down here, you select the layout of your channels. You get a drop-down list, you will see this later on, of all available MEA layouts, and simply you select one from the list, and then accordingly the layout of the data display will be. You get the well-known 8x8 plot over here and one zoomed in channel down there. And one interesting feature here is that while your data acquisition keeps running, keeps showing you data up here, you can pause that zoom channel and zoom in and stuff like that, and then can continue with the live data. Pretty neat. And also over here, that is the window that you get when you have to set the gain manually. You get a list of common devices, and here you type the gain, and that's it. It's common gain for all electrode raw data though. Next in line, the MEA 2100 system. Here the gain is detected automatically. Otherwise, we have much the same functions: sampling rate, the analog channels, hardware info. And here you can see how the drop-down list for the different MEAs looks like. You get even a graphical representation of the layout, so it's pretty easy to find the layout that you're actually using. You select the layout and the display will change accordingly. Then you get the raw data, and down here, that's the zoom in on one specific channel. The data acquisition system has three outputs, as you can see them here. In blue is the electrode raw data. The green is the additional analog channels, the inputs that you have either on your ME data acquisition system or on the interface board of the MEA 2100, if you want to recall those. And there's also the digital channel, shown in red, which either represents internal trigger signals from, for example, the internal stimulator of the MEA 2100, or external inputs from the interface board from the digital inputs on your data acquisition system. This is how it looks like if you add a wireless 2100 system. There it looks a bit different. We still have the sampling rate and the analog channels and the hardware info, but however, here we get the option to scan for available head stages. Then when you did that scan, all available head stages in range show up, and you select one of them, and this head stage then will become active and get connected, and then you get the raw data from this head stage. If you're running the experimenter in a multiple head stage mode, you have to select this here. There will be a short icon indicating that you can switch between single head stage and multiple head stage mode. Once you do that, you will be able to select more than one head stage, and they will simply show up next to each other here. So then you will record simultaneously from all head stages into one file. That's not completely true. When you use more than one head stage, you will get one file per head stage, but they of course synchronize and start and stop together. They can't be controlled independently. So this is now the stimulator interface. That is only available for the MEA 2100 internal stimulator, and in the very near future for the wireless 2100 stimulation device, which is about to come up the next few months. It is not available if you use an older MEA 1060 system with an external STG stimulator. It would be a lot of work to implement the control functions already also in the multi-channel suite. We might do it in the future, but I wouldn't take bets on it. So far, you can control from an older MEA 1060 system, you can control the data acquisition, but you still have to control the stimulator and the selection of stimulation electrodes externally with the MEA Select software and the MC Stimulus software. So how does the stimulator work? It's more graphical than the interface of the external STGs. So you have representations of so-called primitives, so default waveforms. You simply take them and drag them over inside this window here, one after the other, and then you can generate whatever sequence you like. You have three different stimulation lines labeled by colors. Here you have a representation of the available electrodes. You simply click the electrode that you need with the color that you want and assign thereby this electrode to that stimulation channel. You have start and stop functions either for all channels or for only individual stimulation channels. You can start and stop stimulation channel either manually or with an external trigger or an internal trigger. And you have the representation of that specific recording stimulation channel here, and an overlay of all three stimulation channels down there. You can see there are many different waveforms possible. With that icon here, you can even import ASCII files as stimulation files. So you can generate relatively complex stimulation patterns with ease just by a simple drag and drop interface. You click then on the primitive and have down up here the settings for that primitive, and then you go to the next primitive and have again the settings. So it's really easy to use. Then you download everything and you can start straight away. Next instrument is the digital event detector. If I need triggers, I want to trigger, for example, on an incoming trigger TTL from an external device, which is connected to my interface board, to the interface board of the 2100 system. This is information running in the digital channel. I connect this to the digital event detector. In the digital event detector, I have here a simple selection where what are the inputs the event detector should react on. I have here the four dig-in ports on the interface board, for example, and the three internal stimulators, and I can activate or deactivate any of those channels. and I can connect those channels either by AND or conditions, and go either on the rising flank or the falling flank of a TTL. And depending on the conditions that I set here, I get my trigger events, which are labeled with little blue dots here. And the output of that device is a trigger, labeled in pink. Next experiment, pretty easy, is a filter. So you can go with the raw data to the filter, and the output is again raw data, just filtered raw data, but the color is the same, and the functionality is basically the same. On the tab of the filter, you define the filter characteristics. There's a drop-down menu with Bessel, Chebyshev, and other filter characteristics available. Then you define whether it should be high or low pass, the order, and the cutoff frequency. And here in the data display, which is just the same as with the data acquisition, you get the raw data and the filtered data in overlay. So you immediately see what your filter is doing to your data. The output assay is again can be used the same as raw data. The next thing, the next instrument I want to show you is the sweeps instrument. The sweeps instrument is designed to generate data fragments around a trigger event, usually a stimulation. So what you need is a raw data input from the data source to the sweeps instrument, but the sweeps instrument also needs a trigger. So you see that here it has an input for raw data and an input for a trigger. So what I do is I go with the digital data into the digital event detector. The digital event detector looks for the signals coming from the internal stimulator of the MEA 2100, generates up on stimulation a trigger. The trigger goes into the sweeps instrument. Sweeps instrument will generate a data fragment, in this case of 10 milliseconds before, up to 100 milliseconds after that trigger event. And the output is sweeps. It's shown in well, with that color, whatever that color's name is. But from there you can also go into the recorder, of course. You can see here, this is the blanking time, the short time during the stimulation for which all electrodes are switched off. So you have a blank of about 1.5 milliseconds. And right afterwards, there's a very tiny artifact, and just below 2 milliseconds after stimulation, you're ready to record again. Another instrument which also can produce triggers, you can see that here. It has an input for raw data and it has an output for trigger. With this new instrument, you can do something that could never be done with MCREC. And that was one of the problems that could never be solved in MCREC, one of the reasons why we finally came up with the suite. And so we decided to implement this relatively early in the development process. What this trigger generator can do, it can generate triggers, and in contrast to MCREC, the multi-channel suite has a standby mode. So it can go into standby for recording and wait for a trigger input. And this is something that MCREC could never do. So what you can do here, you can go into standby for recording and generate triggers either as a list, as you can see it here, or there can be a periodic trigger, or it can be a manual trigger generated by a mouse click. So you can say something like, give me one trigger every 30 minutes, or one trigger every 5 milliseconds, whatever. Or you can generate a list like, I want my first trigger after 5 milliseconds, the next after 1 second, 5 seconds, 1 minute, 5 minutes, 30 minutes, 1 hour, 2 hour, 12 hour, 24 hours, something like this. Then you will get your triggers. And around each trigger event, the recorder will start a recording for a desired period of time, let's say 5 minutes. So you can build up a rather complex automated recording regime with ease. Next instrument is the spike detector. So like you see it here, you go out with the raw data and either go into the spike detector directly, or first you go through a filter to remove slow wave components. Then with the filtered data, you go into the spike detector. The spike detector detects spikes just the same way as MCREC did. It calculates from 500 milliseconds whenever you hit this estimate button, the standard deviation of the noise, multiplies this with the sigma that you type in here. And for each individual electrode, you get a detection level. And in the threshold mode, everything that crosses that threshold will be detected as an event. Or you can use also the slope mode, just same as in MCREC, combination of slope of the signal and amplitude of the signal. And what you get as an output from that instrument is spike cutouts and timestamps. So you have one data stream, which is just the information about the time of the threshold crossing. And you have another data stream, which contains also a short segment of data around that threshold crossing. The size of these cutouts you can define here. Usually it's a few milliseconds around the threshold. So if you're also interested in the waveform. And this data then can also go into the recorder. The recorder is what we are finally talking about. Everything in the end converges into the recorder. The recorder has a broadband input, which can accept all data streams, of course. Here you see I get the filtered and spiked cutouts and timestamps. I get the direct raw data unchanged. I get the trigger events from the trigger generator and from the digital event detector, all converge into the recorder. You can see here the streams which are being recorded. You can see the instruments they originate from. You can also change the names of these instruments, and they will also change here. If you have something really complex, then you can make your life easier to find your way around. For example, if I have two different trigger detectors, one detecting on the first stimulator and the second only to the second, then you can call that trigger STG1 and the other one trigger STG2. Of course, you don't want to make all these adjustments every time. What you can do, just as an MCREC, you can save this configuration of instruments as a so-called experiment. Up here you see the load and save experiment buttons. And when you need that same configuration again, you simply reload it. In the recorder, I have the option to generate file names with an automatic prefix and suffix. Again, no file will ever be overwritten. If you define a file name which exists just like that already in that folder, the new file will be generated with a name and the ending 001, 002, and so on. The start and stop options for recording are much better than in MCREC. You can start either manually, you can start on an external event or an internal event generated by these devices here, or you can also start on a timer. The same goes for the stop options. Standard function would be, for example, to generate a start event with a trigger generator that goes in here. You select on that event, and the stop you do with a timer, which means, for example, every time the trigger comes in, I record for 1 minute. Then the recorder goes into standby mode again and waits for the next event to come. Okay, anything I wanted to tell you? Okay, starting and stopping, recording, and general data acquisition are up here. This button starts the data acquisition. This button changes between a recording on, standby, and off. If you click the button and the start condition of the recorder is not yet met, you get a yellow light saying in standby, waiting for the right trigger to come. Or if you have the recording started manual, then it will start recording data straight away. So much for the experimenter. Now for the multi-channel analyzer, the offline data analysis package. It supports files generated with a multi-channel experimenter only. And it supports synchronized video input, as I told you already. And it allows you to navigate very easily through the file, either with a scroll bar or simply by digital events that have also been recorded inside the file. And it uses the same simple drag and drop interface as the experimenter. And also custom plugins are possible. Let's see how it looks like. So we have a Spike Explorer and a Raw Data Explorer as, so to say, data sources. I can drag one of them over and have the same tree structure as in the experimenter. What is missing here at the moment is a way to actually record or show the results. That is something that is very high on the to-do list. So what you can do at the moment with you can look at your raw data. You get a representation of one specific channel here. This is the representation of the complete data file. And within that representation of one channel, you can use that blue scroll bar here and scroll back and forth. What you can also do is jump back and forth in certain time windows with that little arrows here. And what you see down here are digital trigger events that also are in the file. And whenever I click on one of those digital events, I will jump immediately to the data corresponding to this digital event. And that does not only go for the electrophysiological data, but also to the synchronized video data. We have here the video player instrument, which can be connected to the data source, and they exchange data. You see that in the form of the icon that data goes in both ways, but here it goes only one way. And when you connect the two, you have the option in the video window, which you can run, of course, in parallel to the data window, to jump to the corresponding data with this button here, and vice versa. From any part of the electrophysiological data, I can immediately jump to the corresponding frame in the video. These windows can be undocked, so you can take them, remove them, and drag them to a different screen, for example, which would be pretty useful if I want to look at electrode raw data and video in parallel. Apart from that, the idea of the analyzer is a bit different from MCREC. What you had to do in MCREC was actually to run through your complete data set to analyze it and also look at it all the time. Much faster than live, of course, but still it took some time for a longer file. This is not the case here. What you do here is you look at a few minutes of the data, you optimize your analysis features, and then you hit the Start Analysis button. You don't see what's happening anymore, but all the data is pushed through that queue in a very short period of time. And down here you will have the output of your results in some way. We are not yet sure how this will look like, but in the end, of course, it will be some exportable data format for the results, be it amplitude, spike, timing, frequencies, whatever it is. The advantage is, of course, that large files can be processed much faster than in MCREC. Last but not least, the multi-channel data manager, the data export tool to third-party applications. As I said, very important. Also, MCREC users only can use this file, can use this software. It will accept all file formats from MCS software, MCD files, multi-channel suite files, multi-well screen files, CMOS control files, whatever it is. You get a representation of the available files in a specific folder here. You have a representation of all the data streams within that folder, and you can export them with a single mouse click. At the moment, we have HDF5, which is compatible with MATLAB and Python. And we also have NeuroExplorer format and Spike2. And there's also a data compression. We will see that a bit larger here. That is how it's a single window. You define the folder where the data comes from, the folder where the exported files should go. Then you select the file or the files that you want to process in a row. The file contents are here, and you can select or deselect all the data streams within that file. Then you hit the export to HDF5, to Next, or to a CED button. You get that window here, and it's processed within a few seconds. Even very large files are processed very fast. That is a large improvement in contrast to the NeuroShare interface, where many people complained that it took ages to convert larger MCD files to MATLAB compatible format. And if you choose the HDF5, there's also a compression level included, which makes sense, especially I think if you are dealing with CMOS data, which can be terabytes actually. Okay, that was basically it. If any questions remain, I will be happy to answer them. Thank you for your attention.
Thank you, Frank. Actually, we already had some questions. First of all, someone was wondering about the systems which work with the software. So we also had the MEA 2100 Lite. Can it also be used with the multi-channel suite?
I'm not 100% sure, but I'm pretty sure it can be. Yeah, let's say yes.
Yeah. Then we have a question about a trigger generator. So could it be used, for example, to avoid recording when the animal is not doing anything with a wireless system?
With a wireless system. That's something we are working on. I think it would probably not work with a trigger generator, but in a different way. However, something that we try to set up at the moment with a wireless system, with a wireless 2100 system to be exact, is some paradigm where the head stage is actually shut down while not in use. So you could do something, or if it works out the way we planned, you could do something like hooking up a motion detector, which then gives you a TTL. And then you say to the software, okay, whenever that TTL comes in, and as long as this TTL is on high, then record. When that TTL is low, then stop recording and go to sleep. Of course, the problem there that we are struggling a bit with is either you simply stop recording, then you won't gain much in terms of battery power, because when you just stop the recording, the head stage still needs power because it still sends data, it's just not recorded. So there's not much use in that. So what you actually have to do is to tell the head stage to shut down and go to sleep. The problem there is when the head stage gets the command to start again, then it needs a few seconds until it's ready for recording, because you have to fire everything up, it has to stabilize. So if you start from sleep mode on a trigger to recording, you will probably lose 2-3 seconds in the beginning, or you have a huge noise, so you can't use a few seconds of data. But that's the trade-off we have to take somehow.
Okay. We had another question to the video player you mentioned. So whether it's necessary to have any other software, or how the video player actually records the image. Okay,
I forgot to tell that. It can be any video format which is Windows compatible. The very last we need to synchronize the video and the electrophysiological recording is one TTL at the beginning and one TTL at the end, and a reasonably stable frame rate of the camera. What is of course better is some internal timestamp in the video file. We had a collaboration with another company where they provided this sort of timestamp, and data synchronization is very precise. However, if you are fine with a few milliseconds up and down, one TTL at the beginning, one at the end would be fine. You have to keep in mind that a standard camera only takes 20-30 images a second anyway.
Okay. Another question was whether there are any pre-programmed experimental setups available for some standard configurations.
No, but pretty easy to do. I mean, you tell me what you need and I can probably put it together in a minute. It's really easy.
Okay.
But we can come up with that. Sure, no problem.
Then there was another question about the spike detector.
Yes.
So if it only detects spikes or also clusters?
Not yet. I mean, there's a primitive spike sorting function included in MCREG. Maybe we will implement that also, or we will implement something else. However, we always have to keep in mind that it might be that we get 256 channels simultaneously. So everything that is really good is usually also very resource consuming. So the question is what we can do in terms of online spike clustering. Offline, okay, that's a different thing. So I think for the analyzer, we will definitely include some spike sorting algorithm. For the online tool, I do not yet know. If we include something, then it will probably not very sophisticated, because we simply lack the resources to do spike sorting on 256 channels at 50 kilohertz while recording data. That's simply brings even the best computer to its limits.
Makes sense. Okay, I think this was it for questions for now. So thank you everyone for attending today's webinar. Well, I think we answered all questions. We will stay online for another 5 minutes or so. So if rethinking everything, you come up with more questions, please again make use of the Q&A window. We have upcoming webinars on some systems which Frank mentioned during the presentation. So our multi-well system and also the wireless system. And as always, we're always happy to answer any question you might have. You see the phone number and email address below. Please give us a call or drop us an email anytime. So thanks, Frank, for the presentation today. And
my pleasure.
Thank you, everyone, for joining today. Have a good day or afternoon. Bye-bye.